The topic that I have been given for today to discuss is about mutational signature analysis, which reveals patterns of genomic instability linked to resistance to endocrine therapy plus minus CDK4/6 inhibitors in hormone positive, HER2 negative metastatic breast cancer. This was originally discussed by Dr. Antonia Mara at ESMO this year. So this is a very interesting topic because we do not generally discuss about mutational signatures, but we know for a fact that because of numerous endogenous and exogenous reasons, there are basically mutations occurring in various tumor cell lines, and this basically agglomerate and develop different mutational signatures. So we have discussed in literature, there are some mutational signatures that have been described, and these mutational signatures might be prognostic as well as predictive, is what we are going to discuss today. So these are some of the mutational signatures, how they develop, and you can see here that there are some mutational signatures like clock-like or the aging signature. This is the commonest mutational signature that we see across all cancers. The second most common is the APOBEC signature. That is basically because of some CT-CG substitution mutations that occurs across cell line, and that leads to the APOBEC mutations, and rest are HRD mutations, MMR deficient, that is immunosensitive, and the smoking related mutagenic signature. So our topic for today is the APOBEC cell signature, and this is more commonly seen in certain cancers like breast cancer, lung cancer, head and neck, cervical and bladder cancers, whereas the clock-like or the aging signature is more common across the other cancers. So which APOBEC signature? So it is mostly the APOBEC 3A and the 3B, which are concerned with the mutagenesis in the breast cancer. They will facilitate the acquisition of mutations which cause progression in breast cancer as well. So the study questions here were, what are the pathologic and clinical features of breast cancer which are enriched for this APOBEC and other mutational signatures? Does it have an impact on the clinical benefit? And what are the genomic alterations which are enriched? So to answer this question, basically we went back to the MSK-IMPACT breast cancer cohort. Basically, this is a clinical sequencing cohort of 4,495 breast cancers with clinical and pathological data being available. And this was basically analyzed for the clinicogenomic analysis, and from that we could get that different mutagenic signatures. And so here you can see that if you go for the whole genomic sequencing, you can actually understand the mutagenic signatures. But if you go for the targeted panel sequencing, or even if you go for the whole exome sequencing, it is not that evident. So you need to go for the whole genomic sequencing, wherein you can understand as to which mutagenic signature this cancer falls into, and there we can differentiate this APOBEC signature from the other signatures. And this again has to be done by a machine learning algorithm. So here there was a sigma score that was used, that is a machine learning algorithm, which basically helps us in deciding as to which cluster of mutagenic signature the tumors fall into. And this was the mutational signature analysis of MSK-IMPACT, where more than 400 genes were analyzed, somatic mutations, copy number alterations, and mutational signatures were derived from the non-synonymous silent somatic single nucleotide variants in samples with at least 5 SNVs. And what was the dominant signature, or the top mutational signature, was basically assessed, and then we were correlating with the other parameters. So you can see here that in the hormone positive metastatic breast cancers, we find more of the APOBEC and the HRD signatures. You can see that both the above two graphs, they are showing that in the hormone positive HER2 negative or the hormone positive HER2 positive, we can see that the APOBEC signature is more and the HRD signature also is more. The HRD is also more in the triple negative breast cancer. Here we can see the genomic evolution in terms of the temporal and the spatial heterogeneity that occurs when we treat the breast cancer, and you can see that the APOBEC actually, it increases once you treat the breast cancer. The APOBEC mutational signature actually accumulates. And if you look at these patients are the ones who have got a high mutational burden and have got a tumor mutational burden is more than 10, and they have got a lobular phenotype, you can see here. And these patients have a poorer progression-free survival on the first-line endocrine monotherapy. So there is endocrine resistance in the patients who have an APOBEC signature. The green line here shows that there is an inferior progression-free survival with the first-line endocrine monotherapy, as well as if these patients receive the first-line endocrine therapy plus CDK4/6 inhibitors, there also these patients are likely to do poorly. And this is why, because of the higher frequency of the specific genetic alterations such as NF1, ARID1A, KM2C, and PTEN, which are associated with the endocrine resistance. So doing a mutational signature will actually enable us to understand which patients are not likely to respond to the endocrine therapy. And if you look at the mutations also, there are some mutations like PIK3CA mutations and the CDH1 mutations, which are higher in the patients who have got this APOBEC signature. Here it is again shown the same, and you might have some double PIK3CA mutants as well as in the APOBEC signatures. So conclusion is that hormone positive, HER2 negative breast cancers acquire genomic instability during the T1 evolution, with APOBEC and the HRD being the main drivers. And patients with breast cancers with high levels of APOBEC are characterized by high tumor mutational burden. Specific histology like lobular cancers, we know that lobular cancers generally do worse than the ductal. And there are mutations in specific genes like specific PIK3 mutations, CDH1, NF1, and PTEN, which also lead to the endocrine resistance. These patients will have shorter progression-free survival on the first-line endocrine therapy and CDK4/6 inhibitor. They also have, even if the patients have a APOBEC dominant or they have a HRD dominant, both these patients will have an inferior progression-free survival. So this is again predictive to the response to the endocrine therapy plus CDK4/6 inhibitor. Thank you.
Thank you so much for this resistance pattern. Now I invite Dr. Surdhana Joga sir, Consultant Medical Oncology, Rajiv Gandhi Cancer Institute, Delhi, for a randomized phase 2b study evaluating chemotherapy combined with ipilimumab and nivolumab IO plus IO in metastatic hormone receptor positive breast cancer.